All right. Last topic of the course, the endocrine system. So the endocrine system, like the nervous system, touches on just about every other body system. It has roles in growth and development, in metabolism, in our long and short-term stress responses, our sexual functioning in terms of desire, our ability to reproduce, our affect or our mood. And so essentially, along with the nervous system, the endocrine system is all about homeostasis. So the first thing we want to do is define what a hormone is. So it's a messenger molecule. And by that I mean, another way of thinking about it is that it's a signaling molecule. But unlike a neurotransmitter, it doesn't act across a tiny distance. It is a molecular trigger that acts far from where it is produced. And because of that, it's a really good way to coordinate the functioning of cells that are physically far apart, whether it's cells, tissues, body parts, etc. So hormones are made by glands, and when we talk about glands, there are two major types, the exocrine glands and the endocrine glands. We're going to primarily be concerned with endocrine glands in these lectures, but I'm just going to remind you a little bit about what an exocrine gland is. In both cases, the exocrine and endocrine glands are modified epithelial tissue. So exocrine glands, like our salivary glands, sweat glands, are ducted glands. So you have hormone-producing cells that surround a central open area. The hormones are produced, and then they are transferred from one place to another through the duct. And that's in contrast to an endocrine gland, which doesn't have ducts, and essentially the blood vessels, it'd be better if this was just laid right over the top. The blood vessels run throughout the gland, and the hormones that are produced go directly into the bloodstream. And that means that any place your blood goes, those hormones can go. So examples of exocrine glands, salivary glands, the stomach, right? The gastric pits have gastric glands, and the pit itself is part of the duct. The exocrine pancreas that produces digestive enzymes and bicarbonate. Sweat glands, the liver is an exocrine gland among its other functions, and the mammary glands of the breasts are also exocrine glands. So this is a much better picture of an endocrine gland. So this is what we're going to spend the rest of our time talking about. Lots of different endocrine glands in different parts of the body, lots of different kinds of hormones that are produced. Because endocrine hormones can reach throughout the body, wherever the blood goes, these hormones go. As I said earlier, they can affect widely separated body parts. And just one good example of that is the hormone estrogen, which is produced by both males and females, although we associate it with females more. The brain, the cardiovascular system, breast tissue, the liver, the reproductive tract, and bone are all affected. They're all target tissues for estrogen. I added this slide just to try to clarify the difference between different kinds of signaling. So autocrine signaling, as you can see in this image, is the cell is producing some sort of signaling molecule, and it may actually have an effect on another cell, but it also has an effect on itself, and that's usually a feedback effect. Paracrine signaling, para means close to or almost. So paracrine signaling is signaling to cells in the immediate area. And then finally, endocrine signaling is signaling where the molecules are transported long distance through the bloodstream to distant target tissues. So one thing that's really important to understand is the answer to this question: do all cells respond to hormones? And the answer to that is no. Only certain types of cells can respond to hormones, and those are what we call target cells or target tissues. And one of the ways I've tried to emphasize this is by having 3 different shaped locks and keys. So any cell that has locks that the hormone key can fit into is considered a target cell. The locks are receptors, which are always proteins. So these are similar to the receptors that we talked about with the nervous system, with a couple of important differences. Sometimes the receptors are outside, facing the outside of the cell. Sometimes they're internal. But in any case, shape determines whether or not a receptor is going to respond to a particular hormone, as you can see. All right, so I've got my little red triangle. Hormone is only going to fit. It's not gonna activate this cell. What you see in the middle panel here is that cells can be targets for more than a single type of hormone. So one question you might ask yourself is, well, what happens if no receptors are present? What then? Or if the lock and the key don't match. And one really good example of that is what happens with Androgen Insensitivity Syndrome. This is a result of a genetic mutation where an XY fetus, right, and normally if you have an X and a Y chromosome, you are genetically male, and your genitals are going to look male, and etc. And if you have 2 copies of the X chromosome, you're female. With androgen insensitivity syndrome, an XY genetically male fetus develops along a female typical pathway. And that's because of a mutation in the code, in the genetic code for the androgen receptors. So although early in development, they're testes and they're producing testosterone, there are no testosterone receptors, or if there are, they're not the right shape, and so the body can't respond to the hormones, and you end up with female typical external genitalia. The person's body, in the absence of androgen or testosterone receptors, converts some of that androgen into estrogen. And so you end up with female typical development. And often women don't know that that they're genetically male because they're only genetically male. In every other way, they're female. There are also some hormone-like messengers, like prostaglandins. They have both autocrine signaling and paracrine signaling. The interferons that we talked about in the immune system chapter are also hormone-like messengers. And then we have molecules called pheromones. Pheromones are volatile organic molecules. Volatile just means evaporate this molecule. It can evaporate easily and become airborne. And pheromones are chemical signals that are produced by one individual that affect the behavior and influence the behavior of other individuals. So many species, their reproductive behaviors are coordinated with these kind of molecules. All right, so we're going to talk about 3 different types of true endocrine hormones. Steroid hormones, which, remember, steroids are lipids. You can always recognize them because they're 4 fused carbon rings. Proteins, hormones, which can be tiny or a lot larger, and then amino acid derivatives. Derivative means you take an amino acid, which is the monomer for protein, and it's modified. So amino acid derivatives are made either from tyrosine, which is an amino acid, or tryptophan. And these can have one of 2 different modes of action. They can either act like a lipid-soluble hormone, like a steroid, or a water-soluble hormone, like a protein. And examples of those are the thyroid hormones T3 and T4, epinephrine, melatonin. You don't need to remember this particular part of it. Certainly you don't need to remember this part. So protein or peptide hormones, if they're something like vasopressin, which is another name for antidiuretic hormone, there are only 3 amino acids, so we would call that a peptide because it's so small. And most of our hormones are, in fact, proteins or peptides. Some, like growth hormone, are ginormous. And in both cases, or in all cases, the receptors for these proteins are on the outside of the cell. Steroids, remember, the parent molecule for all steroids is cholesterol. One reason why it's critical that your liver works so that you can make cholesterol. And the molecular signal for steroids is 4 rings of carbon that are fused together. So estradiol is a really common estrogen. Testosterone is a common androgen. Progesterone, another common female sex hormone. Cortisol and aldosterone are produced in the adrenal glands. All right. Next, we're going to talk about the different mechanisms of action of the 2 different kinds of hormones, lipid-soluble and water-soluble. In this slide, I just want to point out that although the endocrine system may seem simple in terms of, you have hormones and you have receptors and target cells, there are lots of moving pieces. For example, after the hormone is built, it may be stored where it was created or in another location and then to be secreted later. Then it needs to be transported through the bloodstream. It needs to interact with the receptor and then create a cellular response. It's usually that response, as well as perhaps the blood levels of the hormone itself, that have a negative feedback effect on hormone synthesis. And you also have the breakdown of hormone once it's produced. Hormones have a huge variety of effects in our bodies, but the effects fall into three basic categories. Some hormones cause the uptake or the release of other substances, including other hormones. In some cases, they cause changes in the structure of a cell. For example, the structure of tissue, for example, bone. And in other cases, they change the metabolism of cells. The actual mechanism, the molecular mechanism of how a cell responds, depends on where the hormone receptor is located, whether it is facing the outside of the cell, so it's embedded in the cell membrane, or whether the receptor is located inside the cell. And really that is the difference between having a polar, also called hydrophilic or water-soluble hormone, or having a lipid-soluble hormone, which would be nonpolar and hydrophobic. So whenever you see an extracellular receptor for a hormone, the mechanism has to do with the fact that the hormone is unable to get direct into the cell. So it's too large, it's too polar. So the hormone interacts with a receptor that's embedded in the cell membrane, which is sort of a mustard color here. Then you have in the cytoplasm what's referred to as signal transduction. So essentially, for every one hormone receptor complex that's activated, you have a lot of other relay molecules that are activated. And then that leads to the production of the cellular response, but it's indirect and it involves what's referred to as a second messenger. So the next slide, I'm going to walk you through the same process again, but it looks a little bit more complicated. So we have water-soluble hormones, right? They're interacting with a cell surface receptor, and that leads to the activation of a protein called a G protein. And you don't need to remember that to get to AP2. That G protein moves through the membrane, which remember is a fluid at body temperature, and whacks into a third protein that has an ATP bound to it. That turns the ATP into what we call cyclic AMP, which is usually abbreviated lowercase c, and then AMP for adenosine monophosphate. The cyclic AMP activates a pre-existing protein, and then that's what actually causes the effect. If you have a lipid-soluble hormone, like a steroid, for example, which should really have how many carbon rings? Yes, 4. It's my non-happy carbon rings. Right, they're small, and they're non-polar. They go directly slide through the cell membrane, and that's where they bind to the steroid receptor. Sometimes these are inside the nucleus, sometimes they're in the cytoplasm. Another huge difference here is that the steroid receptor complex binds to the DNA and starts transcribing messenger RNA. So you end up with a completely new protein built. Okay, so again, looks more complicated, but same process. Lipid-soluble hormone, right? Doesn't interact with the receptor on the surface. It goes directly into the cell. That's where it meets up with the receptor, which then, translocates into the nucleus of the cell, and then it sits down on the DNA in a very particular spot and starts transcribing messenger RNA, which is then exported to the cytoplasm, and the ribosomes read the messenger RNA and build a brand new protein. So these two differences in mechanism lead to differences in the length of time that it takes the hormone to act. So when you have a cell surface receptor, right, you have water-soluble or polar hormones, they act by generating second messengers, such as cyclic AMP, that then alter the activity of other molecules that already exist. So that means that the mechanism of action is really fast. So insulin and glucagon, which you'll learn about, both act in this way. Steroid hormones and thyroid hormones, which are both nonpolar, interact with intracellular receptors, and they actually change what genes are read. So it takes a lot longer to see an effect of a steroid hormone because you're transcribing and translating the genetic material into brand new proteins. So it takes longer to get turned on, but you're usually changing the structure of the cell, and so the effects last longer. So this is sort of a check for understanding slide. How can you tell what kind of hormone is pictured? So on the right side and then the left side. So there are a couple things to look for. One, you can look for the classic molecular signature of a steroid, which you know is a lipid, which is 4 carbon rings. So that's one thing. You can look at where the hormone interacts with the receptor. Right? If it's inside, it's going to be a steroid hormone. If the steroid receptor complex interacts with the DNA and leads to transcription of messenger RNA and building of a new protein, all of those things separately and together add up to it being a nonpolar hormone, which is going to take longer to begin to act after it's released, but will have an effect for a longer period of time. On the left side, right, we can see that the hormone is interacting with the cell surface receptor, so that suggests that it's polar. We've got adenylate cyclase. Don't worry about that. It's a G protein. But we've got cyclic AMP. We've got an inactive protein that's then activated, which then activates another one, right? So you're not building any new proteins. All right. Next little mini lecture, we're going to start talking about gross anatomy of the endocrine system.